Aliens. What are they? The definition of an alien is something relating to beings from other worlds or an extraterrestrial. This means that maybe life on other alien worlds could look like things that are not just from Earth, but could look like entirely different beings altogether. In this series, we will be creating an alien world from single-celled life to multicellular life that is possibly intelligent. So with this, we will be moving on to the second episode of Project Gibohagg. Now, a popular theory on how life began on Earth is abiogenesis. This is the natural process by which life arises from non-living matter, such as organic compounds. We can have something similar to this happen on Gibohagg too. This is also proven by the Miller-Urey experiment, that came out in 1952, where they simulated the factors of early Earth, like a dense methane and ammonia atmosphere, high temperatures, and they adapted with lightning, and they created amino acids. Some of the amino acids that can form in Gibohagg may start organizing themselves into RNA. These RNA molecules will be capable of self-replication. There's also something called a lipid bilayer. These layers have a hydrophilic head and a hydrophilic tail. These are also capable of self-replication, meaning they can duplicate themselves and mold themselves into different shapes. It is possible that these two maybe can work together. These two may form a sort of relationship with the lipid bilayer covering the RNA. These will form the first protocells of Gibohagg. On Earth, we use deoxyribonucleic acid to store information These polymers are capable of self-replication, development, and gene storage. They are made of deoxyribose, but all methods of storing information do not have to be. There is something called PNA, or peptide nucleic acid. These are also capable of self-replication, development, and gene storage, but these are better at holding polymers together and is more stable than DNA. A peptide backbone and a deoxyribose backbone are also compatible with each other and can form a single strand of storage. This is the process that will happen on Gibohagg. Lipid bilayers can also have different shapes also. It is possible a mutation can cause the cell to have two different lipid bilayers inside of each other. These would make the cell more rigid so it can hold its shape better. In between these two layers, there could be something akin to a vacuole that can form. These would be able to store food in it so that the cell can survive the harsh months without any food or any nutrients going into it. It is possible that the outside lipid bilayer will be able to will be rougher than the inside one. So the inside one is more circular and the outside one is more rough. This would have the added benefit of making it so the cells can stick to rocks, such as the ones on hydrothermal vents. These cells would get their food from hydrothermal vents. These are they're underwater volcanoes that can produce nutrients from the Earth's mantle. They would eat this and just convert it into energy. These organisms would also need a ribosome. These would scan the DNA to make proteins for the cell. These will stick to the inside lipid bilayer of the cell. This is also what cells on Earth do. Now we have a cell. Here's what it would look like in the open ocean. Come back next video to see what prokaryotic life evolved on Gibohagg.